to becoming a biomolecular researcher. I never had any great plan in mind. I didn't know really what I was going to be doing in advance. I just blundered around. I took advantage of opportunities when I found them. I listened to friends and got the advice of mentors as I went, and somehow things worked out okay. So I really want to get across to you in today's talk the idea that you don't have to have a great strategic goal, and in fact, pretty much nobody in science has a long-term strategic goal. They've also just blundered around and things have worked out for them. So who am I? I'm a lecturer in genetics in the School of Life Sciences at the University of Essex. And that means that I teach undergraduates on genetics and related biological science courses. But it also means that I run a research lab in the school. So we are conducting scientific research, trying to explore the boundaries of human knowledge and to learn new things ourselves about how the world works. And it's a really cool job. It has its tedious moments, like all jobs, but the idea that we are exploring the frontiers of human knowledge is incredibly exciting. It's a great motivation to get up in the morning. So what's the kind of research that my lab gets up to? Well, basically, I am interested in understanding how DNA is packaged up inside a cell nucleus. And there are lots of cell types in which this packaging of DNA is really important. The picture here on the left is showing us an egg that is being fertilized by sperm. And you can see just at first glance, there's a huge difference between these types of cells. An egg is absolutely massive, whereas the sperm are really, really tiny. And that means that the DNA that they contain is packaged very differently. It's a lot looser inside the egg. It doesn't need to be very compact. But inside the sperm heads, that DNA needs to be wound up really, really tightly and really carefully packaged so that it can fit into such a small space. And once you start looking at these ways in which DNA gets packaged, you start to ask a lot of questions about how does that packaging occur? How do cells regulate this process when sperm are being produced? Does it matter what piece of DNA goes where inside the head of the sperm? And if this process goes wrong, what kinds of impacts are there on fertility and on general health? So these are some of the questions that I'm really interested in trying to answer. And in order to do this, what I have been spending a lot of time on is developing software tools that let us take images of sperm and automatically measure shapes. So we can measure the shape that DNA occupies inside a sperm head. And we have been applying this in various different species, mice, pigs, humans, for which there are various different questions to be asked, from the fertility and infertility treatment questions in humans, through to improving agricultural fertility in pigs and understanding evolutionary questions using mice. There are loads and loads of different questions, all using the same sort of approaches of studying images and doing image analysis. But that's where I am right now. The path that I want to outline to you in this talk today is how I got here from a position that's not too dissimilar from a position that you might be in right now, because it has in no way been a straight and obvious path to working on this kind of DNA packaging story. It's been a very long and very wibbly road to get here. So I started on this path, I guess you could say, back when I was making my decisions as to which A-levels to take. So that is me back in around 2002 when I was starting my own A-levels. And after GCSEs, there were kind of two directions that I could have gone down. Either the science direction or more the history direction, because I'd enjoyed both of these kinds of topics at GCSE. And I was toying with the idea of going down doing A-levels in history and English, or maybe going down the science and maths routes. And in the end, I decided to do science and maths, but it certainly wasn't a clear decision for me. It was a little bit of a flip of the coin to choose what A-levels I'd actually do. But having made this decision that I wanted to do science-based A-levels, I then picked biology, chemistry, and maths. And I also did Latin because I'd done this at GCSE, kind of enjoyed it. It was an extra thing I could do. So I had these biology, chemistry, maths A-levels that were going on and which made some kind of sense to me. I was enjoying the work. I found it interesting. But the next question is, what do I do next? Where will I go in life after school has finished? And my parents hadn't been to university before, so they couldn't really give me any advice on this. A couple of my aunts had been to university, but they didn't really have any specific advice that they could give me that I found really helpful. So university wasn't a guaranteed thing. It was only because I got some advice from careers talks and these kinds of things that were organised within the school that I had the idea that maybe university would be a possibility for me. And my predicted A-level results were high enough that I could consider A-levels as a thing. But that didn't really help in choosing what course I might want to do at A-level. I'd really got to this level of analysis when I was thinking about what I was doing. I'd enjoyed biology and I kind of enjoyed chemistry, particularly the organic chemistry modules that I was doing. And I hadn't really enjoyed the maths as much, certainly not the pure maths and the statistics. That was all a little bit too complex for my tastes. So when it came to making applications for university, I decided to just take the easy option and split it down the middle. I thought, let's apply for some biochemistry courses because that's going to be a bit of biology and it's also going to be a bit of chemistry. So I don't have to think about things too much right now. I can put the decisions off into the future for a little bit longer. So I decided I would make applications to university for biochemistry degrees. And when I looked around the universities, there were lots of possibilities. I went to several different open days, and I decided that my first choice university would be Bristol, which had a lovely biology department. They seemed very friendly down there, and they had requirements for AAB at A-level. So I thought, this is great. I will go down to Bristol. I will live life in this new city. I will explore all the wonders of a biochemistry degree down there. And then I actually did my A-level exams and I got a BBC. This, you might be able to figure out, is not AAB. So I didn't meet the requirements that were needed to get into Bristol. And so I spent A-level results day sitting in a bit of a funk and feeling rather sorry for myself and realizing that my grand plan of going to Bristol and just doing biochemistry wasn't going to happen. I was going to need to change my course somewhat. And then the day after this, something happened that really cheered me up. my second choice university, the University of Kent, gave me a phone call. And the admissions tutor at the University of Kent, a guy called Peter Nichols, gave me a call and said, well, we saw from the exam results when they came out that you're not going to be going to your first choice, but your grades are certainly good enough to get into the University of Kent, who required, I think, BBC. And and he said, we'd love to have you come join us. It would be great if you come here. That was really friendly of them. That cheered me up a great deal, the feeling that they might actually want me, or that someone might actually want me after I had failed to get into Bristol. So I thought, yeah, I'm going to go to the University of Kent. This is somewhere that seems to want me. This could be quite fun. And so that is the choice that pushed me to go to the University of Kent for my undergraduate degree. And so this is what I did. I went off to the University of Kent to do a 3-year biochemistry degree. And university was a fantastic, confusing and terrifying experience, but also incredibly exciting. I was dealing with moving out from home, living on campus in my first year, and then a shared house in my second and final years. I was learning how to adult, do all the things that come with being an independent person living on their own. But also I was being exposed to this academic environment that I had never seen before. Now, when I was at school, I was absolutely one of the nerds that aren't the cool kids. I was one of the people who got bullied and spent break times hiding in the library because it's where the cool kids wouldn't be found, and I could spend my time reading books and learning about things without being pestered. Once I got to university, I started to find that there were a lot more of these kind of folks around. A lot of the other people just found learning stuff cool, and so started to make some friends that I'd just never really had at school before. And there were so many opportunities. There was this huge library filled with books and journals to go explore. There were lectures going on, not just the lectures on my particular course, but I was able to sneak into some of the other lectures for modules that I wasn't taking, just hang in the background and listen to all of this cool stuff that was going on. It was a fantastic opportunity to learn things. And so a great, exciting time with all of this change happening in my life. But after the first year of my undergraduate degree, we all went home for the summer holidays, and my parents decided that I wasn't just going to spend the summer lounging around doing nothing. They decided if I was going to be home for summer, I was going to be paying them some rent for my board and lodging. And in order to cover this, I would need to get some kind of temporary job. And so the temporary job that I found was with HMRC, Her Majesty's Revenue and Customs, the tax office. And this lovely building in the centre of Maidstone is the offices for the tax office in Maidstone. It was not a fun experience. I realised that working in an office is an utterly tedious, horrible, boring thing to do. It gave me a huge amount of motivation to do basically anything other than work in an office. And so it particularly encouraged me to come up with some kind of a plan for the summer of my second year that would mean I'd get my costs covered, but I wouldn't have to go work in an office. And so in my second year, I was quite keen to talk to some of the researchers who had labs in the biosciences department and see if there's any way I could do some work with them over that summer rather than have to go back to the HMRC. And so this is what I did in the summer of my second year. I got to work in one of the research labs, and we were looking at the development of fruit flies, in particular their salivary glands. So these are fruit flies. They're a particular species of fruit fly called Drosophila melanogaster, and they are a really common model in biosciences for investigating how organisms develop from embryos through to adult organisms. And so my undergraduate supervisor in this project was interested in salivary gland development. And so I got to play around with some really cool biological tools. I got to learn how to breed fruit flies, how to cross different genetic lines together to find results, how to identify males versus females, and how to dissect out salivary glands and to do some really cool microscopy on them. And I realized that this kind of research is a lot of fun. Poking around doing science was something that really appealed to me. And so I stayed in that lab for my final year research project. Because during the final years of bioscience degrees, what usually happens is that every student gets assigned to a research lab. They get to do a proper research project, something that is really geared to helping the work the lab does, but also teaches you some scientific techniques, lets you do your own little bit of research. And so I stayed in that same lab for my final year research project, knowing that I really wanted to stay doing research after my undergraduate degree had finished. And I asked this guy if he had any PhD positions going so that I could continue working with his group and doing more research. And together, he and I applied for a bit of funding that would have paid for me to be able to do a PhD. But that funding didn't come through. And so I wasn't going to be able to stay with his group. I was going to have to move on and to do a PhD elsewhere if I really wanted to do this. And so I wasn't happy. I was disappointed I wouldn't get to carry on down this really cool topic, but I accepted it. I figured I'm going to have to move on somewhere else to do a PhD. So while I was preparing applications for PhD positions, I figured I should get some advice on my CV and get it up to scratch. And so I went to talk to my personal tutor, and all students get assigned a personal tutor, someone who can help them with things like tarting up their CVs. And my personal tutor, I had never met before because he had just that year moved down to Kent. This was a guy called Darren Griffin. And so I went to Darren's office to ask him about my CV. And Darren gave me some really great advice. He seemed like a friendly guy, very helpful. And after talking to him about my CV, my interests, and why I wanted to do a PhD, he came out and asked me, "So, are you interested in working on chickens?" Chickens. I liked chickens, but I'd never particularly thought about research on chickens before. And what he did was offer me a PhD position in his research group that was just establishing itself at Kent to work on genetics and evolution of chicken chromosomes and do some comparative work seeing what kinds of rearrangements had occurred in chickens versus other bird species. So this is a completely different direction to the developmental biology that I'd been excited about and had been planning. But I wasn't going to turn him down. I went to meet the other members of his research group, and they seemed like friendly people. They seemed like people that I could get on with and have fun with for a few years while I was doing my PhD. So eventually I said yes, did the formal application, and got accepted onto a PhD program with Darren's group. And so suddenly I found that I was doing genetics and evolution now, looking at bird chromosomes. What I learned is that chromosomes are incredibly cool. So this is one of the microscopy pictures that I took during the course of my PhD. Each of these little fuzzy blue blobs is a chromosome. You can see we've got some big chromosomes here. We've got some absolutely tiny chromosomes around here. And so what I was doing in my PhD was comparing the chromosomes of chickens and ducks and some other birds, trying to ask, are the bits of DNA in the same order on a chicken chromosome as we see them on a duck chromosome? I was mapping little bits of DNA. Now you see these little red dots here and here at the end of a particular pair of chromosomes. I was hybridizing these little probes onto chromosomes and mapping what order they were found in the chicken. And I hybridized them onto duck chromosomes, asking, are they in the same order? And this showed me that microscopy really is very, very cool. You get these beautiful pictures. And it was one experience in particular that has always stuck with me. It was a late night. I had a whole load of slides that needed to have images taken at the microscope. And I'd been working really late. I think it was about 10 o'clock. Everyone else in the department had already gone home. I was the only one in the entire building. I was taking all of these pictures. And most of them were showing absolutely no difference between chicken and duck. But then one particular probe, one particular set of slides, I took the pictures and there was a difference. There was something different between chickens and ducks. And as I took those pictures, I realized that I was the first person in the entire world to have learned this new fact about chickens and ducks. It may seem a tiny thing, but just realizing that I had learned something that no one else had ever learned ever before, and I was now going to get to tell them all about this cool thing that I had just learned, was incredible. It was a fantastic feeling. It made the late night really worthwhile. After that, there was still loads more slides to take pictures of, and so I finished up, left the department. I think it was raining, walked to the bus stop and realized that I'd missed the last bus because it was so late and had to walk all the way back down the hill to home. But it was worth it. It was such a cool feeling, realizing that I was learning things for the first time that no one else had ever known. So PhD was a really great time. I was making friends. I got opportunities to visit other countries, visiting collaborators and going to conferences and making cool contacts. And I was lucky in that Darren's group was quite large. That's me there at the back. And we were a close-knit group. We all hung out together. We were going to parties and barbecues around each other's houses. And so we were having a great time. And I'm in touch with many, many people from that time. So Darren and Kate, who's next to him, we're still in very close contact and still working on things to this day. So it came with these brilliant opportunities. These are some pictures I took from a conference that we went to in Romania. This is Transylvania, the beautiful mountainous region in Romania. And the conference itself was being held in the capital city in Bucharest, about a couple of 100 miles away. But the people hosting the conference put us onto some coaches on the final day and took us out to Transylvania so that we could see some of the tourist attractions. One of which is this castle here on the left-hand side, which is the castle of Vlad Tepes, also known as Vlad Dracul, or popularly known as the inspiration for Dracula. So this was Dracula's castle, which when we went around was really, really cool and very subdued. It was just done up as a stately home with no real no real signs that this was the famous Dracula Castle, until we got out the final door at the end into the gift shop, which was just packed full of tatty Dracula merchandise, keychains with fangs on them, and all this kind of fun stuff. But on the coach, on the way back, I was sitting next to someone and having a chat about science, because we were all from this conference. And I realized after a few minutes of talking to this guy that this was one of the big names in the field. This was someone whose papers I had read, whose research I had admired, and now I was getting to just chat to him on a coach about science and other cool things. And he was thinking that some of my ideas were interesting. Again, this was incredibly exciting and gave me a great feeling about research. I realized research really is something that I wanted to continue with in life. But eventually a PhD comes to an end after 4 years of doing this work and writing up my thesis. I submitted it. I did the oral examination, the viva, that comes at the end of a PhD, and I had to decide what would come next. And so I knew that I did want to stay in research, so I needed to find a postdoctoral research position somewhere. And I had a lead through Darren who had some contacts. Particularly there was a guy in Cambridge called Nabil Afara, who was a on and off collaborator with Darren. And I'd met Nabil a few times at some little conferences, and he seemed like a nice guy. And Darren told me that he had a grant that had just been funded that had a postdoc position on it, and encouraged me to talk to him and apply, which I did. And so this led me to my next position. This is the Department of Pathology in Cambridge, so Cambridge University. Not somewhere that I had ever in my wildest dreams considered that I might get to work at. I'd failed to get into Bristol. There was no way that I would be good enough to work in Cambridge ever, but somehow here I was getting to work in a place like this with all of the history and knowledge that comes with it, all of these smart people working on cool projects. So I had joined Nabil Afara's group, and that's Nabil right there. He was, again, another really cool supervisor. He was gregarious, he was fun. You always knew when he was walking down the corridor because you could hear him laughing and having a chat with someone. And so, again, in Nabil's group, I met some other great friends and worked on some really cool topics, because once again, the topic had shifted. I was no longer working on bird chromosomes. Now I was working on pigs, and particularly the pig sex chromosomes. Because whenever you sequence the DNA from an organism, you need to be able to assemble that DNA into complete chromosomes. And this is a painstaking process, and sex chromosomes particularly are difficult to sequence. And we're talking here about the X chromosome and the Y chromosome, which some of you may have heard of before. And so this project was a shift from the microscopy I'd been doing before. I had to learn a whole new set of techniques about how you sequence DNA, how you assemble chromosomes. And so I was learning more about chromosome structure and evolution. And we put all of these things together and started to learn what order the genes are on the pig Y chromosome and on the pig X chromosome. But at the same time I was doing all of this, Nabil offered me some experience at doing a bit of teaching. He said, would you like to give some lectures to undergraduate students? And well, my initial reaction was, oh my God, no, that sounds horrifying and terrifying. I can't do this. But when he asked me, we were in the pub and I'd had a couple of pints. And so I didn't say, oh my God, no. I said, okay, that sounds like it could be fun, and set myself up for this terrifying teaching opportunity. And I was only 4 or 5 years older than these undergraduates. I thought there's no way that I could possibly have anything worth telling them. But I stressed about it, panicked, cobbled some lectures together and delivered them. The students seemed to enjoy them. I certainly didn't seem to break them too badly. And I realized that teaching was also quite fun. It was quite satisfying being able to tell people things and help them understand things. So these taster lectures made me realize that maybe this could be a long-term path. From Nabil's perspective, that canny old devil had managed to offload some of his teaching onto me, which saved him a lot of time. So that was pig sex chromosomes, but while I was doing my research, there was a guy working at the desk next to me, Peter Ellis, also in Nabil's group, who was working on mice. And we started talking about the sperm of these mice that he was working with, because they were pretty cool. This is a normal mouse sperm, in case you've never had the good fortune to see one before. They've got this really cool hook-shaped structure. This is the sperm head, and the sperm tail would be coming off down the bottom here. And normally you'd get 50% males and 50% females when a male fertilizes a female and has offspring. But with a particular genetic mutant that we were working with that had deleted 2/3 of its Y chromosome, there was an interesting shape abnormality. These sperm were blunted in their head shape. It's like they run face first into a brick wall, rather than having that smooth curve that we see in normal mice. And they also produced more female offspring than male offspring, which is incredibly interesting. And so we really wanted to understand why they have this difference in their offspring sex ratio and what's going on with this weird shape. And so this led us to a question of how do you actually go about measuring shape? How do you put numbers onto shape? And so I took another turn in the techniques that I was studying and started to learn computer programming so that we could write a tool that would let us measure shapes. And it took a long time. But 5 years after we first started thinking about this, I had written this software tool that would let us do image analysis and automatically measure shapes and measure particularly mouse sperm shapes. And what we did as a result of this research was prove that sperm that carry a Y chromosome are a little bit wonkier in their shapes when they have this YQ deletion than the sperm that carried an X chromosome. It was a really, really subtle difference, but it very nicely explained why males with this Y chromosome deletion had more female offspring than male offspring. It was really beautiful. And we only finally published that a couple of years ago. So this was cool, and it was another direction that I never expected to take. But my contract on my postdoc work was running out. These are short-term contracts. They only last 3 years each. And so the question was, do I try to stay as a postdoc and apply to someone else's group, or do I move on and try to advance my career? And I realized that at this point, I was getting quite expensive as a postdoc. I'd done a few of them, and now my salary was getting towards the upper end of the pay scale. There were people coming fresh out of PhDs who were a lot cheaper than I was. So really, I needed to move on. Also, I had a lot of ideas of things that I wanted to investigate, and I didn't have enough time to investigate them all for myself. So I needed to start my own group. I needed minions who I could dispatch to investigate interesting topics, students of my own who I could train up to use these techniques I'd been developing, and investigate these interesting questions that I had. And luckily, I had an old friend, Radu Zabet, from my PhD days. We had been friends back at the University of Kent while I was doing my PhD. And he now worked at the University of Essex and told me that Essex was hiring new lecturers. There were applications opening up very shortly, and said, you should apply for this. And I said, oh my goodness, this is terrifying and stressful. Am I really ready to run my own research group? He said, just apply for it. So I did. I put in an application, and I applied in around February of 2019, and they held interviews. So I went down to Essex for a daunting interview panel that ran for a couple of days, where I had to outline my plans for my research and how I could see myself fitting in with the teaching and research environment of the university. But I was one of the people who was recruited. And so in July of 2019, I joined the University of Essex, and I got, for the very first time, my own office. Now, this may look particularly exciting to you. It does look a little bit shabby and run down because I'd just moved in. This was taken around Christmas time, but it was my own office. I had my own research group. I was able to start building towards my own little empire in the department. And then everything changed. In March 2020, we all went home, and I have not been back to my office since. I've been working from home for longer than I was ever in my office at Essex. But the research has continued. I have been able to take on my own PhD and master's students. I supervise undergraduate projects in their own final year project, as I was once supervised, and I teach on several of the genetics modules in our various life sciences degrees, which has been incredibly satisfying and very rewarding. It's been a lot of fun. And I continue to work with loads and loads of people. So Darren and I are still working on various projects that have spun out from the work I did during my PhD. Peter, who I worked with in Cambridge, has since gone on to start his own research group, and we work together on mouse stuff all the time. Kate, who was another one of Darren's PhD students while I was there, is also running her own research group and is a lecturer at another university. We put in a grant together to do some pig-based research last year. We continue to collaborate. So all the way through this process, I have been working with lots of old friends and continuing to use these contacts to do cool stuff with cool people. So if we come back to this slide that I showed you at the very beginning, what do? I investigate how DNA is packaged inside a nucleus, and I develop software tools to measure shapes in images. I hope that you can see that that is nowhere at all where I started from. It is in no way the interests that I had when I first went to university, and I never could have conceived in my wildest dreams that this is where I would be if I'd thought about this 10, 15 years ago. So really, it's not about having a plan. It's about trying to recognize when there are opportunities available and take advantage of them. Make friends, use mentors and people that you like hanging out with, and take advantage of the opportunities that they offer. They will tell you cool things. They will push you to do things that you didn't dream that you could do. And above all, enjoy the journey. I'm here because I think research is awesome. I love doing science. If it's something that you find yourselves loving to do, maybe this could be a career for you as well. So that is really all I wanted to tell to you. I think Katie wanted to mention some of the the next steps that are available if you're thinking about university programs and what we have at the University of Essex. So I'll leave this slide up and see if there are any questions that people have got. And thank you for listening.
Thank you, Ben. I just wanted to quickly say as well that that was incredible. I obviously haven't seen your presentation before, and it was really nice to find out about your journey. And like Ben said, if you have any questions for Ben, please pop them in the chat, or you can raise your hand to verbally ask those questions as well. But I just wanted to highlight the next opportunities that we have available at the university. So we currently have a podcast. It's called Understanding University, the Ultimate Uni Podcast, and we talk a bit about student finance, student life, how to budget, stress busting tips, which you can find on SoundCloud by searching for Understanding University. We also have lots of other life sciences talks that are currently available on our website to celebrate British Science Week. So if today was something that interested you, you can find out more about different topics within life sciences and our other science areas on our schools and colleges page. And if you have any further questions from today, you can talk to our students and staff on our UniBuddy platform. And all you have to do is search for University of Essex chat in a browser, and it will come up with all the profiles for you to ask questions. But you have got quite a few questions there, Ben, so I'll leave that to you. But one last thing is that we have the opportunity for students that are interested in finding out more about the University of Essex to complete a little online form, which I'm just going to pop into the chat. If you're interested and you would like more information from the University of Essex, please complete that with your details. And there is a email subscription which you can opt out of, so that you are not bombarded with emails if that's not something you want. But I'll leave it to you, Ben, for questions. If you need me to read them out, let me know.
Yes, I can't see any of the team's chat while I had the screen share on, so okay. I'm just going to stop that screen share so I can see things again. So Enrique, what has been my favorite project so far? This has probably been the sperm-shaped software development, because this was something completely alien to what I'd been thinking before. It was such a new set of skills that I'd learned, but it built on the interests that I'd been developing from microscopy during my PhD and the structure of DNA and how it is organized. It was building on these, but in an entirely new way. And this was a project that I conceived. I wasn't doing someone else's work. This wasn't a project that had been given to me where I sort of implemented their plan. This was something where I thought, maybe we can do this. And I took it right from the first principles to actually getting a final result. So that was my favorite. Andrea's saying, so I created this tool. Was this my first time working with programming? Pretty much. I did no programming at all during my PhD. Once I became a postdoc and started working with DNA sequencing, I started to learn that genetics and genomics is incredibly data intensive. There's huge mountains of data. And in order to make sense of it, we have to use some degree of scripting or programming. So I'd been starting to learn some of the basic tools for that, but nothing to the extent of writing my own software. So this was a learning process. Basically, I taught myself programming by making this tool, which meant I made a whole bunch of very silly mistakes along the way. But it was great motivation to learn. knowing that I wanted to achieve this task kept me at it, when if you're just trying to learn programming with no goal in mind, you might easily just say, oh, this is hard, I'm going to stop now. I think having that project really gave me motivation. And what do we got? Chanel, pros and cons of studying abroad. Oh, that's a good one. I think a huge pro of studying abroad would be that you're getting to experience an entirely different culture. You're going to meet people from very different backgrounds and potentially get to work with people and on topics that you might not have the opportunities for where you're from. The cons are that it's an entirely different culture and people from a different background, and that might be intimidating and uncomfortable. Maybe people in the place that you go to have a culture that you don't find you enjoy, but you won't know until you go. And you will certainly learn something valuable about yourself that you will bring back for the rest of your life. Are there any other questions in there? Do I have missed?
I have a quick question for you, Ben. I have a few that I may have time to ask. And I think Enrique might be typing something as we speak. I wonder if you work with anyone who followed a straighter path than you did into the area that you're working in. Super interesting to hear about all the things that influenced the journey that you took to get where you are. But do you work with people who had a clear goal that they want to work within a particular aspect of genetics or biomolecular research, who have followed that in a more linear way than you did?
Very, very few. It turns out that it's quite difficult to have a predetermined goal in mind and then stick to it, just because the way life throws its slings and arrows at you. You get accepted into this, you don't get accepted into that. And I think having too firm a goal in advance will limit you, because you won't have the flexibility to recognize opportunities when they arise.
This is very interesting, and I find this particularly interesting because I know a few of the students who are listening to this have a sort of primary goal in mind at the moment, which is to study medicine, which is a goal which is very difficult to achieve, and one where, like it or not, disappointment is quite likely and probable. And as part of your story of your journey to where you are now, your very successful place that you are now, Ben, you've encountered a few kind of major disappointments, really. One, not getting into Bristol that you mentioned, one, not being able to continue with your undergraduate project. And these are kind of potential stumbling blocks that would really throw some people off pursuing this kind of pathway. And I wonder, beyond the advice that you've already given, if you have any kind of, resilience is a kind of real buzzword in schools at the moment, but I wonder if you have any advice for these students who are likely to encounter some kind of decisions that don't go their way, to kind of be a little bit more resilient in the face of some of these decisions.
Yeah, I think what helped me when I had these setbacks was friends and family. The support of other people was really, really powerful in making me not feel like I was worthless when I'd had these rejections. And they pushed me to keep on applying for things, even when I kind of didn't want to. When I wanted to sort of curl up under the covers and just say, no, I'm not going to try this again because it was painful when I got rejected. And they were right to do so, because eventually I was accepted onto things.
And before we kind of change direction slightly onto Andrea's question, which we will come to, just on a kind of similar note, I think it was really interesting for me to hear about these points where the journey of your research really changed. You know, you'd immersed yourself in something that you'd become super interested in, and you'd kind of thought that this was something that you'd spend more time in, and then suddenly you found yourself doing something completely different. I think it's interesting that you were able, and I think this is a sort of interesting story for the students to hear at this stage, that at this stage they think there is this pathway that they absolutely want to commit themselves to, but actually there will be these junctions that they encounter that lead them into something else. I wonder if anything kind of particular to you that meant that you were able to also enjoy these new pathways, or if you have any, I don't know whether if you have any kind of, if you're able to share some of your experience about what meant you were able to enjoy these different pathways as they arose.
I think I have always been interested in lots of things. So right from the time when I was at school, I was always in the library reading books on pretty much everything. But what I've also found is that things that I wasn't initially interested in have become interesting simply by virtue of me investigating them in this much detail. So one thing that I often tell my undergraduate students when they're looking for PhD and master's positions is, don't worry too much about the topic. It will become interesting to you just because you've immersed yourself in it so much.
That is a great bit of advice, actually. There was something else that was just on my mind that I was going to ask. Oh yeah, some of your decisions were driven by meeting people who you got on with. Mm-hmm. And I wonder how important you would think, or you know, how important is that to the choices that you make?
Utterly vital. Now, I've just said that the topic you work on becomes interesting to you, which means the topic isn't that important. What's important is that you're with a group of people who will support you and who you like. Because when you're in research and when you're doing PhDs, you spend so much time with them. If you don't get on with these people, if they're not going to be a good supervisor, a good mentor, a good friend, then you're not in a good environment. So having those friendships and working with the right people is the most important part of it. And what I see running my own research group is that being a good manager is more important than doing the top-notch science. Supporting my students and making sure they feel comfortable and encouraged, that's what really matters.
And I think it was really interesting to hear about your experience with your university choices and university application. And it was the university that reached out to you and made you feel wanted and welcome that became the beginning of this pathway that you embarked on. And I think that's something important for our students to think about at this stage as well, that maybe when you get your results, it's not necessarily the kind of prestigious university that you anticipated you'd be going for, but there's real value in choosing the university that looks after its students. Yeah, and we do have a lot of students who end up with very positive stories like the one that you experienced from University of Kent. We should probably answer Andrea's question.
Yes, of course. Has my career opened up many opportunities to travel to different countries? Oh, yes. So there have been many conferences over the years in various delightful parts of the world. One brilliant opportunity in 2016 was a trip down to Brazil, and that was part of a conference series. Now, the previous conference in that series was held in Canterbury in 2014, and I'd given a presentation at that conference, and I won the Best Talk prize. And the prize for the Best Talk was they pay your airfare to go to the next conference in Brazil. So that was absolutely brilliant. I would never have been able to afford to go to the conference otherwise on the budget I had available. And so I got to go to the conference and do a bit of sightseeing at the same time. So these opportunities are out there. They're a great perk of science. Not quite so much at the moment, of course. We are swinging a bit more towards virtual conferences as we start thinking about the impacts of climate change and not trying to jet off around the world quite as much as we used to. But they are out there. And you also get to visit collaborators in other countries and do these knowledge exchanges that are another chance to spend a bit of time in another place. So yeah, science is quite a good career for getting to see other countries. And if you're not going to them, people from other countries are coming to us. We have students and collaborators from all around the world in Essex. So we still get to be exposed to different cultures, different ways of thinking by virtue of being at a university.
All right, I think we should probably finish up there, unless there's any last minute questions which are going to come in, but I don't think there will be. It's been incredibly interesting to hear about your journey into the pathway that you ended up in, and I think all sorts of really valuable lessons for people at this stage to hear about and to be able to learn from. Also incredibly interesting to hear about what it actually is to work in a research laboratory, and to hear a little bit about your progress from being a researcher through to leading a research team, and the decisions that have led you there and your experience in those places. So thank you very much for sharing that. And thank you, Katie, for your information on Essex. It is really important for the students to experience these universities who are doing a lot to look after students at this stage. So thank you very much for that.
It's been an absolute pleasure to come talk to you today.
Thank you very much. Yeah, all our dealings with Essex University have been wonderful. It's also really nice. I really like seeing the pictures of the campus. The picture that you showed, Ben, of the when you talked about your interview of I don't know what building that was. Maybe it's the research facilities.
It's actually our largest lecture theatre.
Oh wow, gosh, yeah. So absolutely, if you're motivated by studying in these incredible kind of pieces of architecture, these modern buildings, then it seems like a really good choice.
Oh, absolutely.
All right, thank you, both of you. Thank you all the students for attending. Thank you for your questions. And yeah, bye for now.
Cheerio.
Bye. Thank you. Thank you.